Hello and welcome back, Year 12 students. All right, in our last video, we looked at anaerobic fermentation in animals. So the differences here are that it creates a lactic acid as a final output with its glycolysis here, all right, compared to that of yeast, where we have carbon dioxide as an output as well as ethanol, which is alcohol. Okay, and we looked at the differences here. Anaerobic fermentation is going to be much faster, but much less efficient and cannot continue indefinitely. Okay, so you hold your breath and try to swim. Good luck for doing that more than a minute or so. Okay, whereas you can go for a jog indefinitely. Doesn't matter how fit you are. Really, if you're really unfit, you can still jog for a couple of minutes straight. All right. You might be puffing, but you can still go for a 30-minute jog. All right. All right, in this video, we're going to be looking at the factors affecting the rate of cellular respiration. Okay, and we've seen this stuff before, so it's going to be a bit of a quicker one. All right, so again, your main factors are going to be temperature and pH as per usual. In this case, because it's aerobic respiration, we have glucose availability, because it's an input, oxygen availability, because it's an input, and of course, enzyme inhibition as well. Okay, so the way enzymes are affected by different things. All right, again, temperature has this graph where it starts to increase slowly. Well, actually, it's a bit faster than slowly. It's a little bit exponentially, but increase like a J-shaped curve. Then it gets to your optimal point, and then it will have a sharp decline as the enzymes start to denature. All right, so kinetic energy increases, which then means the molecules are firing and hitting each other more and more, and then that starts to taper off as the enzymes denature. Okay. pH is more of a bell-shaped curve here again, with an optimal pH here and then a set amount of pH out of its tolerance range, which will denature the enzymes, and the same on either side. So an increase in pH or a decrease in pH should be plus minus the same before the enzymes denature with pH. Okay, so it's going to be that bell-shaped curve. Glucose availability. All right, glucose is obviously an input for glycolysis. All right, so it's both in aerobic respiration and anaerobic fermentation. Okay. So if you take away glucose or increase glucose, it's going to have an increase here of respiration rate. All right. Until this happens to get to a saturation point, and then the substrate essentially can't deal with it as much. All right. So the glycolysis gets to a maximum rate and it tapers off. All right. Oxygen is similar. All right. As you increase oxygen, rate of aerobic respiration or anaerobic fermentation is just aerobic respiration is going to be increased. All right, so if there is no oxygen, that's where anaerobic fermentation occurs. And again, that tapers off and plateaus up the top here. Okay. And then enzyme inhibition. So what's an inhibitor for an enzyme? It's a molecule that binds to and prevents an enzyme from doing its function. Okay, so the old substrate lock and key active site sort of thing. Okay. To create a product. Without that, all right, either the enzyme is denatured or it's inhibited. All right, so we have two different types of inhibitors. Competitive inhibitors. So they're competitive. So essentially, you have your enzyme. All right, you have your substrate that's going to come in here. It's going to be the same shape, and that wants to be split in half essentially. All right, to create the two products. And you have your competitive inhibitor, which just slots in here and changes the shape significantly. All right, now that shape has changed to this sort of shape, and therefore it can't work. All right, so this is blocked. All right, that's your competitive inhibitor. It competitively comes in and blocks the active site. All right, whereas if I'm to draw this again, same sort of thing this time. All right, this time we have this extra part down the bottom. All right, a non-competitive inhibitor comes along, all right, and we'll do it in red again, and it binds to an allosteric site, which is a site away from the active site, and then that will end up changing the active site. Okay, so now it looks like this, and again, whatever the substrate is that's trying to come in here, so say it was, that sort of shape, it no longer can fit. Okay. So that is your enzyme inhibition. All right, thanks for watching, guys. I'll see you in the next video.